Hello children, today we are going to learn a very interesting part of the chapter, and that is the study of the disease AIDS. All of you must be very familiar with this particular disease, or at least you've heard about it in lower classes. Now what exactly is AIDS? Let us expand the word AIDS. The word AIDS stands for Acquired Immunodeficiency Syndrome. Let us dissect each word and see what exactly it means. The word acquired means that which we acquire in our lifetime. Something that we don't genetically inherit, but we have acquired it in our lifetime. The word immunodeficiency is something that a disease which reduces our immunity. We become immunocompromised, or we become immunodeficient. The patient becomes immunodeficient. Syndrome, the word syndrome means it's a group of symptoms. So that is acquired immunodeficiency syndrome. Now this particular disease was first reported in 1981. So it's a relatively very recent kind of disease, and if you look at the evolution of this disease, let me first talk about the mode of transmission. The causative organism. The causative organism for this particular disease is HIV, or human immunodeficiency virus, or the human immunovirus. This virus was first seen in a group of monkeys where it was known as the SIV, or the simian immunovirus, a group of monkeys which were found in Central African countries. Now probably from these monkeys it has entered into human beings, and this type of transmission from animals to man is known as a zoonotic transmission. So the SIV was transmitted to human beings, and from there it started mutating. SIV is simian. S stands for simian. The word simian means monkey. So SIV entered into human beings and it mutated into human immunovirus that we are familiar with. So this is the causal organism, human immunovirus. This is how the virus looks like, very similar to the coronavirus that we are very familiar with. Now these two green color structures are the two single-stranded RNAs. They are single-stranded, and here you find the enzyme reverse transcriptase. This is the protein layer called as a capsosome, made up of individual units called capsid layer made of individual units, capsosomes, and here is the lipid layer which is derived from the host. This is the glycoprotein layer. The glycoprotein layer contains spikes which are capable of recognizing the helper T cells, the CD4 sites. So this is the structure of the HIV retrovirus. So let us discuss about the different methods of transmission. The first method of transmission, and the very common method of transmission of AIDS, is through sexual contact. Normally this virus is spread from a patient to a healthy person through body fluids, either the semen or any kind of vaginal fluids may cause the spread of disease from a patient to a healthy person. It's a very common method of transmission. The second method is through blood and blood products. Normally when there is a requirement of blood or blood products, as in the case of hemophilia, when constant blood transfusion is required, what happens is, if the blood is not screened, nowadays there are blood banks which screen the blood before it is transfused. However, if blood is not screened before transfusion, and if the blood is contaminated, either blood or blood products like plasma, serum, whatever it is, any of these blood products, if it is contaminated and it is transfused to a healthy person, there are chances for the disease to spread to a healthy person. The third method of transmission is by sharing infected needles. Now, this is a very common method of transmission seen among intravenous drug users. So, there are some drug addicts who inject the drugs into the body through needles, and the same needle is shared to another person. So if the first person is an AIDS patient, or if the first person is HIV positive, there are chances for the disease to spread from one person to another. So any kind of infected equipments, surgical equipments like needles, are a potential threat or a potential way by which the disease is spread from one person to another. The fourth method is known as the transplacental method of transmission. It's a vertical transmission. Vertical transmission means if a mother is HIV positive, there are chances for her to transmit the virus to her unborn fetus through the placenta. That is known as vertical transmission, and the method of transmission is known as transplacental transmission. So these are the four common methods of transmission. However, please understand that HIV is not spread through just physical contact. Merely hugging a person or a casual kiss will never cause the disease to spread from one person to another. There are many other cases also reported which does not fall under this particular category, like tattooing. If a needle, an infected needle is used for tattooing, infected in the sense which contains these viruses, there are chances for the disease to spread from one person to another. Or ears and nose piercing. There have been reports about cases spreading in this method also. However, that is very, very remote. The most common methods are through sexual contact, and that when I say sexual contact, through unprotected sexual contact. When there is sexual contact by using a protective device like a condom, there is less chance of the disease from spreading from one person to another. So that is the first method. The second is through blood and blood products. The third is sharing infected needles, especially common among drug users, and transplacental method. Let us now discuss about the incubation period, or the time taken for the virus to exhibit the symptoms in a patient. The incubation period of the HIV virus may vary from a few months. Okay, sometimes the symptoms may appear within a few months, and sometimes it may be prolonged to even 10 years. So from a few months to almost 10 years, it varies from patient to patient. So after a person is infected, it may appear, the symptoms may appear maybe within 5 years, or sometimes even after 10 years. So it's pretty long duration. But there is one particular time or a period which is known as a window period, and the window period is 2 to 6 weeks. This is a time when the virus cannot be detected in the bloodstream by any of the tests. Like there are many tests like the ELISA test or the Western blot test. So all these tests will not reveal the presence of the virus in the bloodstream, and that period is known as the window period. Let us now discuss about the high risk group. Now, who are likely to be infected with AIDS? There are several categories of people. The first category are those individuals who have sexual contact with multiple partners, either homosexual relationship or heterosexual relationship. When there is sexual relationship with multiple partners And when it is an unprotected sexual relationship, there is a chance for the disease to spread. So they belong to the high risk group. The second one is those individuals who are resorting to drug abuse by using intravenous drugs. So they have a tendency to share the needles and they fall under the high risk category. The third one is the recipients of blood as well as blood products. There are patients who require blood and blood products on a regular frequency, especially patients like hemophilic patients or thalassemia patients. They require blood and blood products. So they also fall into the high risk category. And the fourth category is children who are born to HIV parents. So such children also fall into the high risk group of individuals. So these are the four categories of high risk group. Let us now discuss about how exactly the human immunovirus enters into a host and what happens to the virus within the host cells. The HIV, which is a retrovirus, enters into a new human being through any of the four methods that we discussed earlier, either through sexual contact or through blood and blood products, infected needles, or from the mother to an unborn fetus through the placenta. So either of these four methods enables the virus to enter into a human being. On entering, it first attacks a macrophage. Now this is an enlarged view of a macrophage. So it goes and attaches itself to the macrophage. After which the core of the virus, remember the core of the virus which contains the genomic RNA. The core of the virus is introduced into the macrophage and you'll find the viral RNA now within the macrophage. Along with the RNA, the enzyme reverse transcriptase is also introduced into the macrophage. Now what happens next is reverse transcriptase enables the viral RNA to be converted into viral DNA. So a reverse transcription happens and the RNA is now transcribed into viral DNA. The viral DNA promptly enters into the nucleus of the macrophage and there the viral DNA incorporates itself with the human DNA. So it becomes part of the human DNA. I am sure you have studied that the DNA constantly undergoes a process of transcription where the DNA is converted into RNAs. Now when the human DNA undergoes transcription, the incorporated viral DNA also undergoes transcription by which new viral strands are produced. So the viral DNA continues to produce new viral RNAs. These viral RNAs now utilize the lipids of the macrophage and they are now converted into new HIV viruses. This is then released out from the macrophage. The viral DNA within the nucleus of the macrophage remains there and the macrophage now continues to behave like a HIV factory producing more and more HIVs. The macrophage as such does not die but it is influenced by the viral DNA to produce newer and newer viral RNAs. These viruses, the new HIV viruses which are released out of the macrophage now goes and attaches themselves to the helper T cells. So a large number of such viruses enter and attaches themselves to the helper T cells, and there is a progressive decrease in the helper T cells. Helper T cells are now destroyed because of this attack of the HIV virus. And the helper T cells, remember that the helper T cells actually enable the B cells to produce antibodies. So, if you now think about the condition of the patient, there is a progressive decrease in the helper T cells, and when there is no helper T cells, simultaneously the B cells are not stimulated to produce antibodies. What happens now? The patient has an immunodeficient condition, or the patient is immunocompromised. There is no immunity in the patient, and that has a long lasting effect. So this is how the virus enters into the human being and starts destroying the helper T cells. The macrophage remains there, continuing the process of producing newer and newer HIV. Let us now discuss about the diagnostic tests for AIDS. There are two tests for diagnosing AIDS. The first one is known as the ELISA test. The word ELISA means enzyme linked immunosorbent assay. Now this particular test shows 5 to 6% false results. However, it is a very commonly used test. The second test is known as the Western blot test. Now please remember, till date there has not been any known drug to test or to completely cure AIDS. Of course, research has been going on from various parts of the country as well as various parts of the world. However, in order to prolong the life of a patient, there is a treatment with antiretroviral drugs, but it is only partially effective, as it only prolongs the life but does not prevent the death of the patient. So the best way to prevent AIDS, of course, is to take adequate precautions so that you do not develop the disease. Prevention is always better than cure. Let us now discuss about prevention of AIDS. Now we all know that till date there is no cure for AIDS. So the only way is to prevent the disease. And there is a very popular saying which says, do not die of ignorance. Die literally as well as figuratively. So if we know the methodology of transmission, it is the best method to prevent the disease. Now let us see what are the different methods for prevention. NACO means National AIDS Control Organization, and several NGOs, non-governmental organizations, have brought about various steps to prevent the spread of diseases. Let us discuss how the diseases can be spread. The first method is screening of blood, because if blood is screened for the presence of the virus, then it is the best method to prevent the disease, especially for those individuals who require blood. Blood is required for various instances, not only just for blood transfusion in hemophilic and thalassaemic patients. It may be required post-surgery, or it may be required when there is an accident and a patient immediately requires blood. So at all these times, there are blood banks which screen the blood before it is transfused to a patient. The second way is to advocate safe sex. Now, when a person is faithful to her or his married partner, then there is no chance for the disease to spread. But once a person starts having multiple partners, then there is a chance for disease to spread. So safe sex and using condoms. Now, government has initiated the process of distributing free condoms among sex workers, because sex workers, again, there is a chance for disease to spread from one person to another. So distribution of condoms and advocating safe sex is another method for preventing AIDS. The third way is using disposable syringes, especially in hospitals and clinics. If the syringe which is used for one patient is discarded, nowadays you know that all the syringes which come are disposable in nature, but not so in earlier times. Earlier times the syringes had to be sterilized, autoclaved before it was used for the next patient. But nowadays we know that it is always disposable syringes which is used for every patient, whether it's for an injection or whether it's for drawing out blood for testing. So use of disposable syringes in hospitals, clinics and laboratories. Then controlling drug abuse. Drug abuse has become a menace. So controlling drug abuse, especially the intravenous method of drug abuse, is one very effective way of preventing this disease from one person to another. And last, regular checkups, especially among the HIV susceptible group of individuals. There are high risk group. As we have already discussed, if the high risk group of people go in for regular health checkups, definitely that is one way by which we can prevent this particular disease from spreading from one person to another.